Hello everyone. In this session, I am going to talk about different sources and classification of natural products. As you all know, pharmacognosy subject is blessed enough to have some incredible sources of drugs, which includes various natural and artificial sources. Among them, the natural sources include plant, animal, mineral, and marine source, whereas the artificial source includes different aspects of plant biotechnology, more specifically tissue culture technique. Categorization of crude drugs has been done as organized drug, which is on the basis of cellular structure, and unorganized drugs on the basis of acellular structure. The term crude drug generally applies to the products from plant and animal origin found in a raw form. However, the term is also applied to inclusion of pharmaceutical products from mineral kingdom origin and not necessarily only of organic origin such as kaolin, bentonite, etc. The term crude drug is referred in relation to the natural product that has not been advanced in value or improved in different conditions by any process or treatment beyond that which is essential for its proper packing and prevention from deterioration. In pharmacognosy, organized drugs comprise those crude drug material which basically represent a part of the plant and are therefore made up of cells having definite cellular structure. In case of unorganized drugs, they are having a diverse group of solid and liquid materials which not consist of parts of plant and are obtained from natural sources by a variety of extraction procedure. In pharmacognosy, the crude drugs may be classified according to their different status or orders. On the basis of their alphabetical status, taxonomical status, morphological status, chemotaxonomical status, pharmacological status, etc. So here we first discuss about alphabetical classification, which in normally which is normally includes various pharmacopoeias and their site and their specific pattern for maintaining order and sequence. The crude drugs are arranged on the basis of different languages, for example, Latin or English names. Some of the pharmacopoeias and reference books which classify crude drugs according to their system are as follows. For example, Indian Pharmacopoeia, British Pharmacopoeia, British Herbal Pharmacopoeia, United States Pharmacopoeia and National Formulary, British Pharmaceutical Codex, European Pharmacopoeia, Encyclopedia of Common Natural Ingredients used in drugs and cosmetics. If we consider alphabetical classification of crude drugs, there are several examples we can put. Starts with acacia, with benzene, benzoin, cinchona, dill, argot, fennel, gentian, hyoscyamus, ipecacuanha, jalap, kurchi, liquorice, myrrh, nux vomica, opium, podophyllum, quassia, raulfia, senna. Uncaria gambier, Vasaka wool fat, Iolo beeswax, Jidori. It means we have employed all the letters, English letters, alphabets to arrange the drugs accordingly. Similarly, in the same fashion, it has been put in the monograph also and official text also. Next, moving towards taxonomical classification, which in generally includes the classification on the basis of various taxonomical aspects like order, family, genus, species, etc. The drugs here are classified according to plants or animals from which they are obtained in different phylum, families, genera, species, subspecies, etc. This method of classification is based on the consideration of natural relationship or phylogeny among plants and animals. The crude drugs of plant origin are classified on the basis of one of the accepted systems of botanical classification. A large number of plant families have certain distinguishing characteristics that permit crude drugs from these families to be studied at one single time. Thus, drugs obtained from plants having alternate leaves, fruits that are berries or capsules, in case of Datura, Hyoscyamus, Belladonna, and Stramonium, are considered with other members of Solanaceae family. In case of animals, all arthropods are grouped, as are all mammals, fish, and other phylogenetic types. This system of classification is criticized for its failure to recognize the organized and unorganized nature of the crude drug. At first sight, this classification looks appealing, but many drugs are not entire plants and represent parts of the plants that have been processed systematically. Furthermore, the system fails to take into an account of chemical nature of active constituents and therapeutic significance of crude drugs. So I have put an example of belladonna, the scientific name of belladonna, Atropa belladonna. The concept came from the taxonomical classification of Atropa belladonna. Similarly for Ocimum sanctum, commonly known as Tulsi. But if we try to divide the entire thing, analyze the entire thing, then it appears to us like, for a particular example, I can put an example. Glycyrrhiza glabra. Glycyrrhiza glabra or Glycyrrhiza glabra, it's commonly known as liquorice. So in that case, Spermatophyta is the phylum for the plant itself. Angiospermy is the division. Dicotyledons coming under the class. Rosalis coming under the order. Leguminosae belongs to the family. The subfamily will be Papilionaceae. The genus will be Glycyrrhiza and species will be Glycyrrhiza glabra. So that will be the scientific name glabra. So Glycyrrhiza glabra. It means all the parameters, phylum, division, class, order, family, subfamily, genus, species, these all are coming all together under one umbrella, that is taxonomical classification of food drugs. Then moving towards morphological classification, which involves uh, the specific group under a certain category. On the basis of the part that you are using or the whole product as well, it depends which is going to be used as organized or unorganized form of drug. Most importantly, the crude drugs are arranged in such a manner that they can be easily classified in different categories like organized drug and unorganized drug. But in case of organized drug, we can divide itself like the leaf portion of the plant, the flower, the fruit, seed, wood, bark, or any other parts like roots or rhizomes. They all are having their own characteristics, and on the basis of those botanical characters, we have to identify the product accordingly. On very first observation, we have to identify the product morphologically, physically, by our naked eyes, by touching the product. These are the actual parameters to be considered in this case. In case of unorganized drugs like dried latex, gums, extracts, etc., we can observe all those products, and by their character we have to identify morphologically accordingly. So if I put the example like seeds of Nax vomica, it's having almost cellular structure, flat cellular structure. Leaves of senna, very thin type of leaves of senna. Rhizomes of turmeric, that is very well known. Popularly it is known as haldi, as you all know. But if we consider part wise that I have mentioned in case of uh, organized drugs, then seeds of Nax vomica, leaves of senna, barks of cinchona, woods of sandalwood, roots belongs to the plant category of Rauwolfia, rhizomes in case of turmeric, flowers in case of saffron, fruits in case of coriander, entire drug like ephedra, dried lattices like opium, resins and resin combinations like balsam tolu and benzoin, dried juices like aloe, red gum, gums like acacia, dried extract like gelatin, agar. The system of classification is more convenient for practical study, and especially when the chemical nature of the drug is not clearly understood. Next, moving towards chemical classification. Chemical classification are divided into different groups according to their chemical nature of their most important constituent or active constituents. Since the pharmacological activity and therapeutic significance of crude drugs are based on the nature of their chemical constituents, it would appear that chemical classification of crude drugs is the preferred method of study. The crude drugs containing alkaloids are grouped together regardless of their morphology and taxonomical relationship. If we put some valuable examples in case of digitalis that containing the group of glycosides as per their character of their its main chemical constituent or active constituent, for example digoxin present in digitalis and it's a glycoside, so that is why it is coming under the plant is coming under or categorized under the group of glycosides as far as the chemical classification of food drug is concerned. Similarly, for opium, it belongs to the character of alkaloid. In case of black catechu, it belongs to the character of tannin. For peppermint, it will be volatile oil. For castor oil, it will be lipids. For acacia, it will be carbohydrate or derived products. For colophony, the characterized uh, category will be uh, resin and resin combinations. In case of shark liver oil, it is coming under vitamins. Insulin coming under the hormones. Casein, gelatin, papain, trypsin, these are all are coming under proteins and enzymes. The crude drugs belonging to the different morphological and taxonomical categories may be brought together provided there is some similarity in the chemical nature of active principles. Means it refers to uh, the statement that on the basis of morphological and taxonomical categories, what I have discussed earlier, that is quite similar along with the characterization of chemical properties of the drug that we are going to categorize under chemical classification of food drugs. Next, I will discuss about pharmacological classification. So in case of pharmacological classification, uh, the subject pharmacology, it's a vast one. So it involves the classification according to the pharmacological action of their chief active constituents or their therapeutic uses. This classification process is of greater importance, and therefore uh, it is subdivided in different categories. The system of classification, uh, regardless of morphology or taxonomical status or chemical relationship, the drugs are grouped together provided uh, they exhibit similar pharmacological action. Thus, cascara, castor oil, senna, jala, colocynth are grouped together as purgatives or laxatives because of their common pharmacological action. Similarly, uh, gentian, cinchona, nux vomica, sweatia are grouped under bitters. The drugs differing in mechanism of action but with the same pharmacological effect are grouped together also, like bulk purgatives, irritant purgatives, emollient purgatives, for etc. Some of the drugs could be classified under two pharmacological headings since they exhibit two different actions. For example, cinchona is classified both under as antimalarial and bitter tonic, and subdivision categorize uh, of pharmacological classification. If I discuss, it starts with drugs acting on GIT. So in this case, uh, gentian drugs like gentian, deal, kurchi, senna, liquorice, they are all are having uh, positive therapeutic effect on gastrointestinal tract in their own way, in their different aspects of mechanisms. Drugs, those acting on respiratory system, for example, Vasaka, Datura, opium, ephedra, tea, etc. Drugs acting on cardiovascular system, digitalis, cinchona, ergot alkaloids, rauwolfia, etc. Drugs acting on CNS, for example, ephedra, datura, belladonna, opium, coffee, etc. Antispasmodics, opium, curare, datura, hyoscyamus, etc. Under the category of anti-cancer, podophyllum, taxus, vinca are important to mention. Under the subcategory of anthelmintic, it will be quassia, bidang, male fern, etc. Cinchona and artemisia is having uh, antimalarial properties, whereas coca is having local anesthetic properties. If I talk about antirheumatics, then aconite, colchicum, and gugul definitely will be the option. Astringent, the example for astringent containing drugs like myrobalan and black catechu. In case of skin protectant, it will be olive oil, cholu balsam, peru balsam, etc. So this is the last category of classification of food drugs, and that is simply referred to chemotaxonomical classification, where uh, we can say it establishes a relationship between different positions of the plant and attempts. Position of the plant basically refers to the uh, position of the plant that has been taken in the plant kingdom, and attempts to utilize chemical facts for more extracts means it is again related to the chemical nature, understanding of the biological evolution and relationships. The characters more often studied in chemotaxonomy are secondary metabolites, which are of greater importance medicinal value, pharmaceutical significance, such as alkaloids, glycoside, flavonoids, etc. For example, the ideal example will be, I guess, the location of berberine alkaloid in Hydrastis, barbaris, and argemone, or we can put an example like distribution of routine, the entire group, etc. Thank you very much.